My lab really is a very kind of interdisciplinary space, if you like. We have three kind of major foci for what we're working on. So one of them is microscale tissue engineering or organoid development. That includes both the biology, but also the development of supporting technologies. At the moment, we have a particular focus in type 1 diabetes. The technical aspect of that is the AgriWell platform that I developed a while back, but have been working on expanding and scaling up and scaling out for both screening and eventual clinical translation applications. And then the biology that's tied to that around type 1 diabetes is the development of these engineered pseudo-islets that would be transplantable to treat type 1 diabetes, although we're using them in, I don't know, probably 20 different tissue types right now in various different collaborations. The second component is revisiting a little bit how we do science. So I feel like we have a lot of technical innovation in terms of what we use to do our science, but sometimes it's maybe overlooked a little bit, this idea of going back and revisiting philosophically how we're doing some of these things, and also just, you know, additions of controls and the whole issue of reproducibility and repeatability of our work. We do both the biological aspects of organoid and microtissue development, but also the development of supporting technologies. So the AgriWell platform is one example there. That's, you know, commercialized and pretty widely used out there in the world right now. I think that we just cleared 1,000 references in Google Scholar database where people mention using the AgriWell platform. But there's also a number of other applications of the toolkit and equipment that we've accumulated to pursue that research. So for instance, working with colleagues in veterinary medicine to use 3D printing to develop teaching simulators, for example, including the oxygen monitoring hardware and oxygen control hardware, again, that we've been using for our organoid development, but it's started to show some pretty clear applications in areas as diverse as in vitro fertilization, and a bunch of different other organoid types that my veterinary medicine colleagues are working on. Because my actual faculty position is in the Faculty of Veterinary Medicine, there's a lot of interactions between the needs, the problems that exist in veterinary medicine, and then the kind of solutions that biomedical engineering is able to provide. There's a lot of different interactions at a lot of different levels, but in some ways, probably the most valuable has been the interactions with other faculty, with people in engineering. I'm on the Foothills campus, and so if I was not involved with biomedical engineering, both the research strategy and then the kind of graduate program that's very tightly associated with that, and of course undergraduate as well, I really wouldn't have much of a profile over on the engineering side. And coming out of that, there have been, you know, of course, a number of collaborations, but also really kind of key student recruitment. I've had some students that have applied to biomedical engineering graduate program and then come to me through that. And also I had, you know, as one specific example, Doug Condro, my recently graduated PhD student, came out of the undergrad biomedical engineering specialization here at U of C. His project was scale up of this AgriWell bioreactor, which I think would have been a challenging project for someone who didn't come in with an engineering background. But he was sort of ideally suited to do that. And that now, you know, has been quite a successful project. That's something now that in kind of the short to medium term, Doug has gone to a full-time position at Stem Cell Technologies in their Cambridge, UK facility, you know, taking development of that microwell bioreactor forward. So putting that platform in place and allowing some translational impact. And that's also really a kind of a keystone for my own grand research vision of establishing a pipeline where we can translate a number of these different microtissue generation strategies or organoid generation strategies to clinical applications. And so by kind of bringing in the engineering component, by working with my colleagues here in VetMed who you know, may have access to animal tissue and things, really integrating that together has really strengthened our ability to push that project forward. There's also accomplishments from other students that recruited through the biomedical engineering grad program, and various collaborations. There's a group of us, myself and some other people, looking at some medical device development, and currently just kind of we're working with Innovate Calgary to go over what the IP landscape is like and whether there's patentability there. And that again is a project that I don't think would have seen the light of day without the BME research strategy.